Hey everyone, and welcome back to Load of Scit. It's been a while, but we are back, and with all new videos. Today's video, mutations. Now, mutations often get an interesting connotation when we think of them in movies. We often think of this, or in my case, even this. However, mutations in themselves are easy to understand. A mutation, by definition, is any error in the genetic code. So your genetic code is a sequence of DNA and RNA and all of the nucleotides that code the proteins that make up who you are. Now, a mutation is an unintended change in this code, and these changes come in a vast array. There are two different classes of mutations. There are point mutations and chromosomal mutations. Point mutations fall into three subcategories: substitutions, insertions, and deletions. Now, the names are pretty straightforward. A substitution is when you substitute one nucleotide, A, C, G, or T, for a different nucleotide. This point mutation can have various genetic effects and are often the cause of single nucleotide polymorphisms. Next, we have insertions, which the name is also pretty easy with this one. It's where we insert a nucleotide that wouldn't be there. We also have deletions, which is when you remove a nucleotide that shouldn't be there. You notice the yellow arrows that appear showing not just a mutation at one point, but across an entire reading frame. We actually call insertions and deletions frame shift mutations because they alter the reading frame of amino acids. Next, we'll move on to larger chromosomal mutations, which names for this one are pretty easy. Inversion, if you can probably guess, is when you take a piece of a genetic segment and flip it around. This can cause various genetic effects based on if it splices genes. We have deletions, which we essentially delete part of a chromosomal segment. We have duplications, which, you guessed it, we make a copy of a chromosomal segment, which can produce even more genetic material, proteins, from those coding regions. And we have translocation, where we take genetic material from one chromosome and reattach it to another chromosome. If a mutation occurs in a nucleic acid's coding regions, or exons, then will generally have a much larger effect on the proteins encoded by that gene. If it occurs in an intron or non-coding region, we generally see that the effects will be less severe on your survival. However, this is what contributes to the differences in between individuals, as I mentioned. So we might be asking ourselves, why will we get mutations? Shouldn't our cells be able to prevent this from happening if our genetic material is so important? Unfortunately, we don't always have a choice when it comes to mutations. Spontaneous mutation is essentially an error in the machinery that copies our DNA polymerase, or RNA polymerase, which copies our nucleic acid so they can be further used. When this happens, I say that it makes an oopsie in that it messes up a letter. Now, our cells are as human as we are, and they can make mistakes. And this includes the polymerases, which do have a high mutation rate. However, DNA polymerase generally tends to fix its errors, and they also have more proofreading mechanisms than RNA polymerases. But RNA are generally more short-lived than DNA. Your DNA is more permanent. Once it reproduces, if there's a mistake in the DNA, that mistake is permanent. So DNA tries to fix itself much more. However, there are some types of mutations that are induced by other means, and we call these, like I said, induced mutations. An induced mutation is also what it sounds like. An outside source will actually interfere with your DNA copying and cause a change in bases. This is the reason why smoking is said to cause cancer, because smoking actually affects your cancer-preventing regions of your DNA and can therefore weaken your ability to fight off cancer. So, mutations are generally classified into two areas based on where they happen in your body. Somatic mutations take place in your body's cells, such as your hair, your skin, your other organs, and germline mutations take place in your gonads, the region where you produce gametes. Now, somatic mutations generally are not passed on to offspring, but because germline mutations take place in the gametes, they can be passed on to your offspring. If an organism's offspring acquires a negative mutation or a deleterious mutation or even just a neutral mutation, you'll generally find it disappears from the general population because it's of little use. However, natural selection actually favors mutations that help out that organism. If two organisms with beneficial mutations come in the population that actually helps them survive better, then you'll find that they'll actually be more fit for reproduction, and they will increase in amount in the population, while the other organisms will either diverge into a new species or the species itself will evolve. And this is actually the basis for evolution, because since natural selection favors beneficial mutations, a species is given the ability to evolve. So this has been a review of mutations, from the different types of mutations to how they're caused to what effects they can have on populations. As you can see, they're not just stuff from science fiction, but they're actual stuff that we can study in labs, and they're the basis of many new frontiers of science. And with that, this has been Loda's Scit, and I'll see you next time.